So this is on all kinds of RNA formulation and diagnostics related topics. And I think we have a very, very good and powerful selection of presentations and topics here in the session. I should be probably the first speaker, and I don't know. Now you should see my screen. Okay, so I decided to give a kind of a broad overview over the field and the somehow coherencies between past, present, and future. And I will talk a little bit about the delivery technologies on LNPs and beyond, before and after. This doesn't work. I know what it is. Yeah. So that's just, let's say, a reminder. These people are now publicly known, and you have seen and heard them quite a lot in the media. So I don't need to say so much more on them. Ugur and his wife Özlem and all the other people of the board who helped us to arrive on where we stand. This is just a reminder on the COVID vaccine mode of action. So we load messenger RNA coding for the spike protein of the COVID virus to nanoparticles, which are delivered to antigen-presenting cells. The RNA is translated into the peptide, and this is then presented. Where is my? Yes, presented by the antigen-presenting cells, and it induces the immune response. So that's what we now know all. And this is also from our company website, where we claim this is a 10-month journey, which is in fact true. So just, let's say, from the kickoff to the final product, we did it at that time. But however, there was, of course, also a life before. And this is why we were able to do all of that. And just to remind you, I've selected one slide from a recent publication in ACS Nano, where people depicted the evolution of nanotechnology, where maybe discovery of the liposomes is one hallmark in the 60s. And then several years later, the first liposomal drugs came up, several of them. There were all the gene therapy applications and several other nanoparticle products. And in parallel, also lots of research going on in this field. And this is basically one of the reasons why we and others were able to do so. And to follow up with that, this is a little bit from my field. That's publication from Cyrus of India from the 90s, last century. That was a period when people investigated rather thoroughly biomembrane systems from a physical perspective. And they had this nice cartoon in their paper, which basically, at least to my knowledge, for the first time transported the concept that DNA at the time, DMA and lipid membrane aggregates, they form staggered multi-layers with a certain type of order, which can be readily investigated by X-ray diffraction. And many parameters, many characteristics can be revealed by doing such studies. And in fact, in the past, lots of people who do biophysics and biomembrane sciences studied these systems very thoroughly and gave a very broad understanding of the systems. And at least from my perspective, this was a very important basis for us when we started formulation development for the systems, and to try to have a directed rational formulation development for the formulations. And now coming to what we are talking about right now. That's again from us. So in addition to the COVID vaccines, of course, we have other projects. As probably you know, by our DNA, we are more a cancer company than a vaccine company. But also the cancer pharmaceuticals, they are vaccines, because we do cancer immunotherapies. And besides, we are now exploring more or less a very broad spectrum of therapeutic options which you can apply on the basis of RNA. This is just from an older slide with some selections, but we have now many different potential applications, therapeutic applications for the RNA currently ongoing in development. And this is just the simplest possible way to depict what we do and how we do. So to have a pharmaceutical compound on that, you need to make nanoparticles, or that's basically what most people do. And in the simplest case, you just combine the RNA with a vehicle, which can be a liposome or something. You end up into the nanoparticle, which is then manufactured as a pharmaceutical product and injected to patients. Then the RNA is delivered to the site of action. And the next slides, I would like to go a little bit through technologies which we are applying right now to tailor and to engineer such products. Here, just maybe for the reason of given interest on LNPs, in that slide, I have compared what we call lipoplexes, and lipoplex is also the term originally used by the earlier biophysical studies, and LNPs were we have both these technologies, but they are just names, if you wish, and if you want, you can call LNPs lipoplex, and you can lipoplexes LNPs. But in our internal wording, we would declare lipoplex as systems which we obtain from combination of RNA with liposomes, so an aqueous mixture, and we result in lamellarly organized systems pretty similar to the ones we have seen on the slide from the earlier publication from the Sardinia group. And you can do it with relatively straightforward combinations of lipids. Optionally, you have a cationic lipid, optionally an ionizable one, helper lipids if you need, and that's all about it. So it's really straightforward. The LNPs, in contrast, they also, first of all, are consisting of lipids and RNA, but you mix the lipids as an ethanol solution. Where in addition to here the ionizable lipid, which changes pH as its charge as a function of pH, you have quite a lot of cholesterol in all known LNP formulations, I know at least. You have a little bit of phospholipid, and in all cases also I know you have a grafted lipid, like a pegylated lipid, which is required for particle engineering, because without that the particles would precipitate, and also to modulate PK after injection. So this is more or less the technology space, and you can now tailor your formulations depending on requirements for a pharmaceutical application. You need tools to investigate the systems, and we like to use quite a lot methods for structure analysis, like X-ray scattering. Here you see X-ray scattering patterns from lipoplexes. You see nice peaks from LNPs. A similar peak, but it's way broader, and this gives us information on how they look like internally. And now I would like to give you just some snapshots of what can be done. So this is just an overview, a collection of methods you use in order to tailor formulations for a given purpose. So you do fundamental investigation of properties like binding studies. You add liposomes to RNA and look at what happens. That's binding. This is X-ray diffraction. This is analysis of X-ray diffraction. There are other methods. And then you can outline regions where the particles, the nanoparticles have certain characteristics, both physicochemically and biologically. And by that, you can, for example, identify formulations which target a certain organ. Another example, going to the world of ionizable systems, you can study again a structure of lipoplexes model systems which comprise cationic or ionizable lipids, and you can then track what happens on changing the pH in order to track the evolution from being injected in circulation at pH 7 point something to endosomal uptake with decreasing pH. And you can reveal structural changes which depend on the composition. I don't go into details, but cationic lipids, for example, don't change their structure because they don't change their charge. But if you have ionized lipids together with RNA, you see structural changes, which then you also correlate to certain model assumptions. And this can be the basis of optimizing pKa behavior for targeting a certain compartment in addition to chemical synthesis, as shown here in the example by Peter Callis' group when they synthesized lipids and did studies with silencing by siRNA. Just another thing, what currently is ongoing. This is this grafted lipid story. As said, we do need these grafted lipids to allow particle engineering. We can use them to adjust the size and then also to adjust circulation time. We use pegylated lipids with that, but in the recent years, more and more concerns came up. First of all, PEG reduces the cellular uptake because it makes the particles stealthy also for the cell birds to be uptaken. But also there are some immune responses against PEG because we are exposed to PEG moieties from consumer chemicals. And therefore, the activity of the product is impeded, and there can be even severe immune responses up to anaphylactic shocks. So this means there would be an interest of alternative grafted lipids, which have the same positive properties as PEG, polyethylene glycol, but not the disadvantages. There are different options, and here you see one with using polysarcosine, a polypeptide. Just again, a summary showing that you can do like particle engineering, more polymeric lipid decreases the particle size, both with molar fraction and chain length. You can see the effects on structure, and you can also then tune the targeting profile on in vivo application towards different organs by changing the right composition of the grafted lipid. Further application of tailoring and designing particles is shown here. That's a busy slide, but I would put your attention to this rectangle where we depicted our understanding of a nanoparticle, which is kind of composed of a mixture of ordered and loss ordered domains. We see this again by X-ray and small angle scattering. And we could figure out that a certain type of internal organization having both ordered and non-ordered moieties had best biological activity, and this we consider as a potential basis for tailoring novel formulations, irrespective of the molecular basis of the polymer and the cationic lipid or the other moiety. So, and then finally, I would like to look a bit into the future, and here I don't have so much time, but I just would like to mention some hot topics. Some of them will be stressed later in the subsequent talks as well. Thermostability was an important one because all of the difficulties due to the -80 storage of some of the vaccines, and thermostability can be limited either by degradation of the RNA. That's a chemical degradation hydrolysis where you have to find ways how to inhibit or slow down this chemical hydrolysis, or in the frozen or lyophilized state, there is aggregation which limits the stability. And the LNPs, they have a disadvantage in comparison to the Lipoplex systems, which are charged, and the charge gives you electrostatic repulsion. So on the freezing stress, they can't as easily get in direct contact and then fuse. And therefore, one has to optimize the systems. But I would say I'm convinced that in the next months or years, systems will show up which are prone to freezing at -20 and also to lyophilization. Another summary of topics: alternative delivery systems. This could be an own presentation of 3/4 of an hour. I just listed some systems which I would consider of interesting, where I expect novel formulations come up to solve problems which currently cannot be solved by the lipid-based ones: polyplexes, hybridism, as mentioned, silica nanoparticles, exosomes, polymersomes, peptide nanoparticles, and many other. And I really look forward to see more of them in clinical practice and in development. And then finally, what I also consider important is alternative application routes. So needle-free application, oral delivery, nasal delivery, inhalatory. Also here we hear something from the next speakers. And I'm confident that we will have novel improved formulations in the products in the next future, which help particularly application in different boundary conditions like emerging countries and so on. With that, I would like to stop and just say, don't read it too much detail, but I would like to focus on the novel delivery technologies and the insight, the control for future development and next generation RNA therapeutics. I would like to thank here all the collaborators: Mainz University, Leiden University, Tel Aviv, and EMBL at DAISY, Hamburg. And with that, I would like to finish and thank you for your attention, and ask you to ask questions later on in the discussion room. Good. So I hope I didn't exceed my time too much. And with that, you have the introduction to the next presentations. The next one is coming from Cynthia Yu Wai-Man, I hope this is pronounced correctly, who will talk about RNA therapeutics in ophthalmology, transition into clinics. And we look forward to your screen.
Thank you very much for the invitation to present today at the meeting. My talk is on RNA therapeutics in ophthalmology, with focus on the interesting nanoparticles which are making progress in clinical trials and making progress in terms of going to patients in clinic. I have no financial disclosures for this specific talk. So glaucoma is the leading cause of irreversible blindness worldwide. It currently affects 76 million people, and the prevalence is expected to increase to 112 million by 2040 due to the aging population. Glaucoma is a condition whereby you have high pressure in the eye, and this leads to damage of the optic nerve, leading to irreversible blindness. The specific area of research that I've been working on over the last few years has been looking at fibrosis and scarring as the main cause of failure of glaucoma surgery. Currently, just to give you a bit of background, the current gold standards in fibrosis in glaucoma surgery has been the cytotoxic antimetabolites, mitomycin C and 5-FU, which are used because it causes widespread cell death and apoptosis in fibroblasts. But unfortunately, the downside to that, it leads to blinding side effects in patients, namely extensive tissue damage and severe infection. There is thus a very large unmet need to develop a much more targeted and less cytotoxic therapeutic in glaucoma surgery. And nanomedicine is showing a lot of promise in this area of research, and a lot of different types of nanoparticles are currently being investigated, namely lipid-based nanocarriers, polymer-based nanocarriers, drug conjugates, inorganic nanoparticles, as well as viral nanoparticles. One specific nanoparticle which we are aiming to take to clinical trial has been in collaboration with Nanogenics, which is developing Liptide, which is a novel peptide nanoscale vehicle which delivers gene therapy to the affected site. It is made up of 3 main constituents: lipid, peptide, and payload, in a ratio of 1:4:1. It starts with central lipid core, to which we add targeting peptides to the surface, namely in the case of glaucoma, targeting integrin receptors, which are abundantly expressed on eye fibroblasts. And the payload is then inserted within the lipid core for delivery into the eye fibroblasts. Every part of liptide can be customized for purpose. In this case, in terms of a payload, any type of gene therapy, be it siRNA, mRNA, gene editing tools, plasmid DNA, and artificial chromosomes. And the peptide can also be customized for update in different types of target cells, the integrin for conjunctival eye fibroblasts, vascular specific ICAM1 neurotensin receptors, and the lipid can be customized for release once taken in the cells by being cationic, anionic, RNA versus DNA payload, and further customization. And the delivery, apart from the eye, can also be targeted to different parts of the body, be it intravenous, ex vivo delivery, intracerebral, and nebulized forms. This table shows the comparison of the liptide nanoparticles compared to the more traditional liposomal nanoparticles and the viral vectors. The advantage of liptide, which is why we've focused on it for glaucoma, has been that it has a higher specificity than the traditional liposomal vectors, but still a lower toxicity risk compared to the viral vectors. In terms of the preclinical results, we transfected conjunctival fibroblasts, which were cultured from glaucoma patients with the liptide nanoparticles targeting specific genes which were important in fibrosis, in this case, the MRCFB gene. And transfecting in the cells led to a 70% gene silencing of MRCF. And on live cell imaging, when we looked at the fibroblasts post-transfection, the control nanoparticle, the fibroblasts looked in normal morphology, whereas the fibroblasts once transfected with liptide MRCF nanoparticles rounded up in shape and could no longer contract as the normal control nanoparticle fibroblasts on the left-hand side. And we put these fibroblasts in a functional collagen contraction assay, and the gels that had the fibroblasts from the control nanoparticles transfection had significantly contracted the gel, which is on the left-hand side, whereas the fibroblasts transfected with the MRCF liptide nanoparticles had barely contracted the gel at day 7, showing that transfection with the liptide MRCF nanoparticles led to a significant decrease in fibroblast contractility and blocked fibroblast contraction. We validated the results in the rabbit model of glaucoma filtration surgery. We used a white New Zealand rabbit as it is the established model for fibrosis in glaucoma surgery. 100 microliters of the liptide nanoparticles were injected subconjunctivally at the end of surgery. And the primary outcome measure was bleb survival. This graph is a Kaplan-Meier graph showing the bleb survival for 3 different types of treatment groups: the mitomycin C, which is a current gold standard, the control nanoparticles, and the liptide MRCF nanoparticles. And we found that the rabbits that were treated with the control nanoparticles, the black line graph, all had failed by 12 days post-surgery in terms of bleb survival, whereas the rabbits transfected who received the liptide MRCF nanoparticles, which is a broken black graph, or mitomycin C, the current gold standard, the gray broken graph line, had a significant increase in the bleb survival. And on average, the control nanoparticles of mean bleb survival was 11 days. The bleb survival for liptide nanoparticle was doubled to 22 days, and for mitomycin C was 22.5 days. At day 30, the rabbits were culled, and we looked at the histological staining for different markers of fibrosis and found that the rabbit conjunctival tissues that had the liptide MRCF nanoparticles, there was a decrease in scar tissue on picroserous red staining, decreased cellularity on H&E staining compared to control nanoparticles and mitomycin C, decrease in myofibroblast staining on the alpha smooth muscle actin staining compared to control nanoparticles and mitomycin C, and a decrease in collagen on the glomerulus trichome staining compared to control nanoparticles and mitomycin C. And also none of the rabbits showed any sign of systemic or local toxicity after transfection with the liptide MRCF nanoparticles. The eye has the distinct advantage of being a closed organ whereby the nanoparticles can be injected very close to the target tissues. This has several advantages. It increases the bioavailability of the drug to the target tissue, the disease tissue, and also decreases the risk of potential side effects in terms of systemic absorption. And currently, there are several RNA therapeutics which are making progress to even phase 2 and phase 3 clinical trials with potential of moving to clinic for patient benefit. To look at different RNAi therapeutics in the eye, the BF655 from Pfizer in terms of age-related macular degeneration and diabetic macular edema has reached phase 2 trial, and QPI 1007 for non-arteritic ischemic optic neuropathies is currently in phase 2A trial, and also different other RNAi therapeutics for dry eyes and glaucoma are currently in phase 2, phase 3 clinical trial. The next step of the research, moving on from the in vitro and in vivo work I've presented, is now moving on to a phase 2A clinical trial in glaucoma patients. We've already designed a clinical trial, and it will be for 30 patients in terms of assessing eligibility. These 30 patients will be randomized to a 1:1:1 ratio between the current gold standard mitomycin C, low-dose lipid MRTF nanoparticles, and the higher dose of lipid MRTF nanoparticles. The patients will be followed up over a 12-month period, and the endpoint will be read at 1 year. In terms of eligibility, the patients will be patients with primary open-angle glaucoma, having primary glaucoma surgery trabeculectomy and over 18 years old. The primary endpoint will be the percentage of patients with no blood failure at month 12 compared to mitomycin C current gold standard. And the secondary endpoints will be toxicity, both ocular and systemic, intraocular pressures, best-corrected visual acuity, the visual field testing, and the OCT disc retinal nerve fiber layer thickness. So in summary, the eye is a closed compartmentalized organ, allowing local delivery of RNA therapeutics close to target tissues. The local delivery affords increased drug bioavailability and decreased adverse effects compared to systemic delivery of nanoparticles. And currently, the phase 2 and phase 3 trials are completed or currently in progress in different eye diseases, be it the retinal diseases and glaucoma. And several therapeutics are showing promising results for clinical translation to patient benefit. I'd like to thank my lab, my collaborators, mentors, and most importantly, patients, without whom we wouldn't be able to do the research. Thank you very much.
Yeah, thank you very much for this very interesting presentation. As discussed, so we will postpone all the questions to the discussion room. And with that, I would like to go to the next speaker, who is Yasin Ekinci from Paul Institute, actually. And he will speak about a nanofluidic device for reliable and quantifiable diagnosis for COVID-19. So,
thank you for introduction. And which screen you see now?
Yes.
Is that correct?
Looks good, yeah.
Okay, good. Well, thank you very much for the introduction. So I will introduce you a new device. And actually, this is a more general purpose immunosensor that can be used, and we applied this to COVID-19 detection, and I would like to share some results about this. So our aim is developing immunosensors, and the conventional way of doing that, for example, the state-of-the-art or golden standard is the ELISA plate, and it's a cumbersome and long process, takes several hours, a lot of very long incubation times and cleaning procedures. And these ELISA tests, they have very high specificity and sensitivity. When you go to the other extreme, we have the lateral flow assays. They are very simple to use and very fast, but they have limited sensitivity. So our aim is, can we bring the sensitivity and specificity of the state-of-the-art immunoassay tests, but in a much simpler and quick way? And for that, we developed a plexiglass 3D nanofluidic device. And it has high sensitivity and specificity, easy readout with a fluorescence microscope, and it doesn't require extensive lab work, and results you have within a few minutes. So the device looks like that. So you see here the video. So actually what we do is we have different nanoparticles, and there are nanochannels, and these nanochannels have a wedge. So they become narrower. And after this trapping region of this wedge, there is a capillary pump. So actually the whole thing is a two-dimensional capillary needle. You put the droplet, it sucks in, and it can sort different microbeads and nanoparticles. And the advantage is now we can use bead-based immunoassays, and then different sizes can target different analytes. That allows us then the multiplex detection. Now, how can we make such devices in cost-effective? First of all, there are thousands of papers about microfluidics and nanofluidics. But if you want nano-functionality, say feature sizes of a few microns, this is very difficult to achieve, so they are expensive. But here in our device, the high precision comes from the height. And all the lateral dimensions are at least 20 micron. And this is just a thermoplastics. So in our case, it is a plexiglass, PMMA, and we can cost effectively pattern this and upscale that. Of course, there are a lot of tricks went into this device, optimization of the capillary pumps and all the geometry, but also functionalization of the nanochannels, etc. And the difficulty is actually having this wedge, because in conventional lithography, you have the digital, so you have a constant height. And if you want to modulate the height, then this is so-called grayscale lithography, and it is difficult, and we manage that. But then what we do is with the e-beam lithography tool, which is expensive and slow, we make the master. And then from this master, then we make daughter masters. And from these daughter masters, then we use then nano-imprint lithography, which is very cheap. And most of the time until now, we use nano-imprint lithography, but a much simpler and quicker way is injection molding. And I would like to just show that how it works. So actually we did also here, you see the, for example, the critical part is this wedge, and it is over a millimeter size, and we can change the height in a very accurate way. And then from our e-beam written master pattern to nickel shims for injection molding, everything works fine. So we have high pattern fidelity and also roughness of the end devices below 5 nanometer. Now, we did this with different thermoplastics, and here you see plexiglass, and here you see COP, and this is then confocal microscope, and here is the AFM image. And now in these videos, what you see is then we put 500 nanometer particles, and you see that they are trapped at the exact location. And this is a video image showing that actually we can trap 500 nanometer particles easily. And then actually then we can put different nanoparticles. In this case, we put 5 different nanoparticles, and they're all trapped at their specific locations. And now we can use this for multiplex detection. When we started this project, we were aiming for mitochondria trapping, and to analyze their size distribution. It's important for some certain diseases, like Parkinson. But then pandemic came, and then we said, what can we do for this societal crisis that turned our lives upside down? And then we started then using these nanoparticle or microbeads for COVID detection. And we targeted then the spike protein. And now then we take a nanobead, and then we can put the spike protein on top of that. And then we have antibodies from the patient, and then if we have COVID positive, then it will bind, and later then we put then fluorescent detection antibody on top of that, and then we do simple FLISA test on these beads. And then here you see the results, calibration beads, and here different concentration of antibodies, and you see the beads here. And then here, when we look at anti-spike IgG, we have detection limit of about 1 nanomolar. And for many assays, you may find this low, but this is, first of all, a relevant range. Secondly, the limit of detection is limited by the specificity of the anti-spike protein to the antibodies that were available at that time. So we did then tests on about 30 patients, and then we have 100% specificity, and the sensitivity was 95 because one patient we have one false negative, and actually this was the blind test, and after that we repeated that, and this was also positive, and we don't know what went wrong, but already in this very first experiments that we could have more than 95% sensitivity is very good. And then we can do also multiplex testing. What we did is then we did IgG, IgM for COVID, and we put also influenza A because these have similar symptoms. And then we can, for example, if someone goes to the doctor, then doctor can immediately say, not only you have COVID positive, but it is not COVID, but it is flu or something else. Of course, this multiplexing platform can be increased further, maybe up to 100 if we use different colors, different channels, different nanoparticle sizes. And this is really a powerful platform. Now, this was the published work. I would like to share some unpublished work. So until now, what we did is then we take the serum and then we mix the nanoparticles or microparticles, polystyrene particles, and then incubate, wash, and then put into the nanochannels. But this takes also, half an hour or even more. And can we do it better and faster? So what we did is then, first we took the particles and trapped them at the locations, and then dried these devices in vacuum. Now, all you have to do is put the serum and the detection antibodies, and we will able to detect. So this is much faster. And we did this with IgG and IgM. It works perfectly. And you see here, actually, the time-lapse studies. I can show also one example here. Actually, within a few minutes, the signal already saturates and we have a detection. So the whole incubation time is reduced to a few minutes. And in typical ELISA test, this is several hours. And the reason is we have a very high surface to volume ratio. Now, moving on further, until now, then we used what I show. Until now, we used serum. And the problem is in a normal blood, we have the red blood cells, and that makes life very complicated for all possible immunoassays and tests. And then you can use different methods. For example, if you use membranes, they got clogged with the time. And if you use paper, then they might have a specific absorptivity, and you may change the concentration. One thing is our test is not only high sensitivity and specificity, we would like to be also quantitative, and that is challenging. And we don't want any interaction with the filter. And then there are, of course, when you go to the microfluidics, there are many ways that you can filter the serum from the whole blood. But these require complicated microfluidic setups, and this hinders the potential practical applications, right? And what we did is actually, now you see here cross-section of our device, and we have the trapping region here, the wedge. So if we just let the whole blood, and if this channel, the narrowest region, is high enough, then the red blood cells can just go through. So we change this height here. And then here you see what happens, how much. So this is a kind of indication of whole blood and different diluted blood going through the channels. This is a microfluorescence image. So for undiluted blood, it's about 2 micron. And for 1 to 40, it's about 1 micron. So practically, if our channel height is more than 2 micron, we can work with whole blood. This just limits that the smallest bead, polystyrene bead we can work with is more than 2 micron, and this is okay. And here we show then the sub 10-minute detection. So actually what you see here is then we can pre-trap the different nanoparticles, and then we can put the whole blood on that, and then we can immediately detect that. Interestingly, the whole blood looks better than the buffered serum. And this is not exactly we understand, but because there are in the blood, there are also many other stuff ongoing, but at least this doesn't deteriorate the performance. We get similar performance from serum or whole blood. Now to conclude, so we created a device which gives a nanoscale topography with grayscale lithography, and we can make these devices very cheap with injection molding. And we can do what we demonstrated is multiplex COVID and influenza testing with high sensitivity and specificity. We can make this process much faster by pre-trapping the particles in the micro channels and drying it. And we can also work with whole blood. So all these steps drastically shorten the testing time and incubation times. So actually this is, we detected here antibodies, but we can also detect antigens. We are expanding our capabilities to further, for example, we have data already with cytokines. At the moment, we are concentrating on more cytokines, which is very interesting actually for various diseases. And we are also increasing our multiplexing capabilities. And with that, then I would like to conclude. Actually, whatever I presented here, 95% of the work is done by my PhD student, Thomas Mortelmans, and there are many colleagues contributed from Paul Scherrer Institute, University of Basel, and Fachhochschule Nordwestschweiz. With that, thank you for your attention.
Yeah, thank you very much for this really interesting presentation on devices and microfluidics. As said, here we can ask questions in the discussion room later on. So we would now move on to Carsten Rudolph from ETRIS. I hope you are still here. And the title of his talk is Immunomodulation and mRNA-based protein replacement therapy programs for treatment of pulmonary diseases.
Super, Heinrich. I hope you can see my screen.
Yeah, looks good. You just have to go to the presentation mode.
Okay, I think now it works. Heinrich and Beat, many thanks for giving us the opportunity here to present and for the invitation. And Heinrich, you addressed already one topic in your presentation, also maybe changing the route of administration from IV or local administration to inhalation or nasal administration to target the pulmonary tract. And that's basically what we do and what our focus of ETRIS is. And I will present to you today some overview of the technology that we developed here at ETRIS, but also some results of the two major programs that we are running right now. One is addressing an approach using messenger RNA for immunomodulation of the messenger RNA of the lung, and the other one addresses the application of messenger RNA to treat an inherited lung disease for replacing a missing protein. So I think I don't have to speak about messenger RNA and all the great things that you can achieve and address with this modality. Yeah, like I mentioned, we're focused on targeting diseases of the lung with our technology that we have developed. But let me go into some more detail on what we have worked over the last years. And we did some work on basically the two parts which are relevant for a messenger RNA drug, and Heinrich spoke to it already. On the one hand side, it's the drug substance, the messenger RNA itself, but we all know that messenger RNA itself is not very well penetrating cell membranes. So that's why we all have to package the messenger RNA with various systems. And I'll give you some idea of how we do it. But let me start with first part of what we developed at ETRIS, and that's modification scheme, yeah, and I mean, we have all experience from the past last year from the vaccine field that indeed it is very helpful to try to reduce and minimize the immunogenicity of the messenger RNA for successful approval of a drug product. And what we've done, we've also developed a modification scheme, yeah, to really minimize the interaction with the toll-like receptors. That means the innate senses of our immune system, but we do it a little bit differently. So we don't focus on replacing just the uridine with an N1 methylpseudine, but we did it in a way that we just do minor changes and replacing minor fractions of two nucleotides at the same time, yeah, which gives pretty much the same results as you see it from those approved messenger RNA vaccines, but it's a lower degree of modification that we do. But on top, we did also, like everyone is doing, a lot of work on designing UTRs to find, I think, a good compromise on the one hand side on good translatability of the messenger RNA, but I think we all know that at the same time we have to address also the yields during production. That means that you get really good transcription rates at the same time. So we've done a lot of work here. And then we did a lot of work on manufacturing of the messenger RNA. And we developed a technology, a process at Athres, which is an HPLC-free process for an easily scalable to get high yields and very pure messenger RNA. So that's something we've done in the last couple of years. And then we've done also a lot of work on looking into formulations for delivery of the messenger RNA. In particular, we developed also a lipidoid technology with a particular focus on delivering the messenger RNA into the cells of the lungs. Yeah, and here you have to consider that you have another layer of complexity, basically, and that's addressing how to nebulize the messenger RNA into the lungs. And you all, I think, are aware that during the nebulization process, there are shear forces acting on the messenger RNA and the more, I would say, fragile LNPs. And so that's why we've done a lot of work to find a way how to stabilize it. And Heinrich, you addressed this already. We've done work with a kind of stabilizing excipient, yeah, that we found that helps to stabilize and avoid aggregation during the nebulization process. But on top, this also helps really on improving the storage conditions, yeah, of the messenger RNA. So we don't need minus 80 degrees for a more longer storage, but we are fine with minus 20 degrees. And we have already also just an in-use stability at 25 degrees with 4 days. So I think that helps really to make that easier being applied by doctors, messenger RNA drugs. Yeah. So overall, our goal is, and I think we've done a lot of progress here to really work on improving the safety and tolerability of messenger RNA drugs for pulmonary application, addressing our storage conditions for the use of the drug, really to make it as easy as possible for patients regarding the inhalation. So it's very rapid inhalation that we've developed, and together with a high drug quality, of course, of the drug. But let me give you some more background now where we use these technologies and where we are there. And I'd like to introduce to you today a program that we call ETH47, and that targets basically respiratory viral infections. And what is ETH47? So it's a first-in-class early intervention for broad spectrum respiratory viral infections treatment by using a messenger RNA that encodes a type 3 interferon. So what is the role of type 3 interference? And type 3 interference is a subset of interference that are in particular active, at the mucosal barriers of our body. That means in the pulmonary tract, but also in the GI tract. Yeah, and it is the first line of defense molecule that is activated after a virus infection, and it triggers basically through a cascade of signals a very broad antiviral response. And what is important to be considered is that the receptors for the type 3 interference are mostly restricted to the mucosal barriers, but only occur on a smaller subset of leukocytes, which adds a kind of improve their safety profile of using such a molecule. But what we want to achieve with this drug is basically to program the airway epithelium, the lung epithelium, into a very broadly antiviral status, so that it's really manipulating the host, so that we can achieve a mode of action that is virus agnostic, so independent of the virus, so that is active against SARS-CoV-2, but also influenza viruses, rhinoviruses, common cold coronaviruses. We think a great opportunity of developing this drug on the one hand side for the treatment of respiratory infections, but also the prevention of respiratory infections, like post-exposure prophylaxis, but even thinking more ahead also into the prevention of exacerbations driven by viruses for mild and severe asthma. And of course, you can think about a pandemic preparedness drug to bridge for potential future pandemics the time until you have really tailored vaccines available. So we tested ETH47 in various virus models, challenge models. And what you see here is where we tested it in an MX1 congenic mouse model in preventing the influenza A virus infection. Yeah? And so what you see here on the left-hand side is on the one hand side, the body weight, and the loss of a body weight is a clear clinical symptom in mice for the response to the virus, and you can nicely see that we really improve and reduce the weight loss there after we treat the mice with ETH47. But what we think is even more important is that, and that's what you see on the right-hand side, that we can really also improve the survival rate in those treated mice by ETH47. So it very clearly shows that this is really a very strong active activity and efficacy that we see from the drug. But we also moved on and looking into another model for for influenza, and that's a ferret model. And the ferret is somehow more closely regarding clinical symptoms related to us as humans. So they really develop a fever and clinical signs that are similar to humans. And what you can see here also is that on the left-hand side, with the treatment of the ferrets that are challenged with the influenza virus, you can nicely flatten the curve regarding the virus replication here, measured as the TCID50. And if you just focus on the center part of the slide, we can nicely inhibit fever in the treated animals compared to the vehicle control. And you can see very clearly that also the clinical symptoms, which is sneezing and nasal discharge activity. This score is greatly improved with the treatment of ETH47. So it very clearly shows that ETH47 is really active in influenza models. But we also moved on then and had a look at ETH47 activity in a SARS-CoV-2 mouse model, and we used here the human ACE2 transgenic mouse model, which is commonly used in the field. And we did basically the same like we did in the influenza challenge models that I presented to you before. And also here you can very nicely see that ETH47 leads to a very substantial reduction in the virus replication. Yeah, so it's a almost 100-fold reduction in virus replication. And we also see again very nicely improving the body weight of the mice, treated mice compared to the vehicle controls. And again, we can really nicely see that ETH47 leads to very strong survival benefit in the mice. So overall, I hope that you agree with me that ETH47 is very effective against the viruses in these animal models. And we moved this program now through the full preclinical program development, and we target first in human studies by the beginning of next year. But let me have some words also around this ETH42 program, where we develop a new messenger RNA drug for the treatment of a primary ciliary dyskinesia. And as I mentioned already before, it's a genetic disease, and this genetic disease leads to mutations in proteins that are responsible for the beating of our cilia in our airway. So, and if these cilia do not properly beat, the mucus, is not well transported out of the way, out of the lungs, and it leads to mucus congestion, which in turn is then again linked to chronic bacterial infections, which can in the end lead to lung failure. And there's currently no treatment available on the market. And what we did here, and I will just show you some initial data, we tested in PCD patient-derived airway liquid interface cultures that were taken from nasal biopsies. And checked these, and these airway liquid interface cultures are really fully differentiated airways. Yeah, so they are really to be considered like a kind of organ model, for the airways in the culture, and they comprise all the cells that are needed for maintenance of the airways: basal cells, club cells, goblet cells, and most important, ciliated cells. And what you see here on the left-hand side, is that by treating these airway liquid interface cultures with ETH42, we can really restore the structural defect. And one typical binding partner of CCDC40 that is missing in those patients is Gas8. And what you can see here is that really in the treated patient airway liquid interface cultures, we can nicely restore the protein function in the cilia, so which indicates that the cilia are corrected structurally, and not only for Gas8, but there are other binding partners like CCDC39, DNLI1 that are again restored to the cilia. But I think what is more important is really are those cilia also functionally corrected? Yeah, and that's what you can see here in the middle part, and I hope you can see the videos playing. Yeah, and on top you see where it's highlighted control. That's how a high-speed video microscopy movie looks like from airway liquid interface cultures of a PCD patient culture, and you see basically not really any movement. And if you have a look at their ccDC40 mRNA treated culture, you can nicely see here really the beating is back in the cilia. And when you quantify it, you also see that we have a nice beating frequency, which is in the range of normal. Yeah? And then we also moved on and checked in another animal model, because there are no good animal models available, but we used the zebrafish in cooperation with the Susana Lopez from Lisbon. And you can also see here very nicely, if you have a look at the nasal pits, where those cilia are active in the zebrafish, and this is an experiment where the messenger RNA was injected earlier in the one cell states of the zebrafish. You can very nicely see that's the wild type here. And that's the control treated nasal pit, and that's here with the ccDC40 mRNA. You can nicely restore the cilia function again. So I think we are very confident that you can also really use messenger RNA to correct genetic lung diseases, and we are very happy and also moving this program forward to the clinics, but it's a little behind from ETH47, so more towards the second half of next year. And that's basically it. And I'd like to thank you all for your attention.
Yeah, thank you very much, Carsten, for this presentation of this novel administration route, which I really appreciate, as said, and I think we look forward to the progress of your project. We are a little bit behind time, so we have to move on. And the next speaker is Michael Keller from Hoffmann La Roche, and he will be talking about the CMC perspective of mRNA-based medicines. How stable are mRNAs in and outside their delivery vehicle? So we look forward to your presentation.
Thank you, Carsten. Can you see it the right way around?
No, you should swap.
Just swap it quickly. Is it better now?
Yes, perfect. Yeah.
Good, excellent. So good afternoon, everyone. I'm going to spend a few minutes talking about the stability study that we have been conducting on messenger RNA-based LNPs. Our interest is mainly to evaluate whether these therapeutic modalities can be used for some of the indications we are interested in. And as an additional therapeutic modalities, as you all know, has been mentioned many times before, including in the first overview talk, messenger RNA needs delivery. The most prominent approach and clinically validated is the encapsulation of messenger RNA into LNPs. I've just printed that out, this nice summary slide from Schoenmaker and colleagues, which shows the formulation features that govern these three different LNPs, three under the market, one with sRNA, two with messenger, one with sRNA, two with messenger RNA-based therapeutic modalities. One striking feature that we can immediately see here is that the ionizable lipids that are present in these formulations are quite different and also have different pKa. So as a chemist, I'm always interested in structure-activity relationship, of course. And what we decided to do is actually to focus down on these ionizable lipids and screen them very broadly. As we have learned also in the first talk, the current approaches to deliver messenger RNAs are mainly based on lipid nanoparticles. There are other types of delivery vehicles that hopefully will come up in the future, and we will learn more about that. We are investigating some of these different principles as well here in my lab. However, the focus is clearly on lipid nanoparticles now. What we decided to do, we used a commercially available messenger RNA from TriLink encoding GFP. Then we used a constant helper lipid composition and just changed one factor at a time, which is the ionizable lipid, and then looked into the stability profile of these resulting messenger RNA-based LNPs. Here is just very briefly the structure of the messenger RNA. For those who are less familiar with it, you have the cap region, you have the 5' UTR coding region, 3' UTR, and then the poly A tail. This messenger RNA has just underneath 1,000 nucleobases and approximate molecular weight of 400,000 Dalton. We went then into the literature and picked out 5 ionizable lipids of interest that is depicted here and started to manufacture LNPs. This is the resulting physical-chemical characteristics. So we have roughly a 0.1 milligram per milliliter final messenger RNA concentration of these 5 different LNPs. We can do sterile filtration. We can carefully characterize them using agarose gel retardation assays, GLS, zeta potential, all the standard analytics that one uses to describe these particles, and then finally the ribogreen assay for content and encapsulation efficiencies. So let me quickly talk you through the study results. When we analyzed them after manufacturing, we quickly realized that when we changed from the manufacturing buffer to the final PBS, there was an increase in size, as you can see here. All the messenger RNA, as far as it is visible on the agarose gel, is fully encapsulated here. So all is fine. We have a slightly near neutral negative zeta potential for all of the formulation. The encapsulation efficiency was really good for all the ionizable lipids that we have investigated, more than 87% for all of them, which is depicted in this graph. And the recoveries of the total messenger RNA that we put into the formulation and got out in the LNP was between 90 and 100%. Now, what happens over 9 weeks? We looked at that very carefully on agarose gels. Nothing really happens. No free messenger RNA came out from the different LNPs in neither of the 5 LNP formulations after 6 and 9 week storage. And when we looked at the particle sizes and the PDIs, we were actually astonished to see that even at 40 degrees, most of the LNPs were pretty stable in terms of overall particle sizes and PDIs. You can see the red outliners here are the stability points at 40 degrees. So not surprisingly, under stress conditions, actually, these LNPs tend to then, at least some of them, tend to become less homogeneous, but most of them are pretty stable. When we looked at the results of the zeta potential, we could see that some of the LNPs were really stable, but others at 40 degrees, LNP 1 and 2, for example, seem to have released messenger RNA, and the zeta potential has changed accordingly. But LNP 3, 4, and 5 really looked amazingly good over the 9 weeks we investigated at 40 degrees. Now, the encapsulation efficiency are pretty much the same. Not much of a change apart from LNP number 1, but the others were really more or less stable apart from LNP 2 at 40 degrees Celsius. Again, this is stress conditions. So we were surprised to see that from an overall physicochemical viewpoint, these messenger RNA-based LNPs seem to be much more stable than we had actually expected. Also, the results from the free messenger RNA concentrations, not spectacular because they are pretty stable, except for the 40 degree conditions for LNP 1 and 2. And finally, we looked at the total messenger R concentration using the ribogreen assay. And also here we have some outliners, but these were due to experimental difficulties, pretty stable messenger RNA concentrations for all the 5 LNPs over a course of 9 weeks. The variations we observed, actually, we figured out that some of the caps that we used were not properly screwed, and we lost some humidity, we lost some volume there, and that explains also that some of these variables, some of these data points actually varied over a course of 9 weeks, and we have to improve on that. Now, what happens with the naked messenger RNA that is not encapsulated in the LNPs? This is an important control that we run concomitantly. We were surprised to see that, what we then define as stability assessed by the ribogreen assay was pretty good with some outliners. Again, here we had some evaporation that was observed due to untied vials that we used. We have to change this type of vials. But our immediate question was really, is the ribogreen assay giving a relevant readout for the content and for the stability of these messenger RNAs within the LNPs, but also when it's not formulated. So what we actually decided to do, we brought these particles to our biological department, biology department, and asked whether they could actually help us evaluating the biological activity. And this is briefly summarized here. We realized very quickly when we provided the first set of samples, there was 5 days between manufacturing and investigation of the biological activity. And most of the LNPs, with the exception of LNP number 1, actually were not active or very much reduced in activity. So we figured something must have happened, and we set up an additional stability study there, where we looked at the time course of LNP number 1. You can see now we were investigating here only over 4 weeks. You can see a decrease of the GFP expression profile in this particular cell culture, 2D cell cultures that we used. We can also freeze and thaw them over a course of at least 1 week using 2 different chemically stabilized messenger RNAs. But this was only the case for 1 single out of these 5 LNPs, where we could actually see increased stability or prolonged stability, whereas all the others, and I'm just showing one here, LNP2, actually were pretty quickly deactivated within the LNP formulation. There was a continuous loss of activity over 4 weeks as well for the LNP1, but it's still active. All the others were actually from the beginning, after a couple of days, pretty deactivated. So we went back into the literature and we found this nice publication from Pecker and colleagues from Moderna that actually very nicely described that the ionizable lipid can lead to chemical reactions with oxygen and then attacks the messenger RNA and actually forms adducts of lipid-based adducts with the messenger RNA, inactivating the messenger RNA pretty quickly. And this obviously depends very heavily on the ionizable lipid that is being used for these messenger RNA-based LNPs. So the deactivation process can be very quickly, can be a little bit more slowly. It depends really on the type and the nature of the ionizable lipids. So as a conclusion now, what we have seen over the last 9 weeks is that generally from a physicochemical viewpoint, the characteristics of the LNPs, they remain largely the same with variations at 40 degrees Celsius, which is stress conditions. However, there might be, and in most of the cases, is a rapid loss of the bioactivity that we have observed. So we need to work on that, and we need to make sure that we understand what type of ionizable lipids are more prone to react with messenger RNA, and how we can actually mitigate that risk and improve on that to finally, at some point, have formulations that also can be stable at room temperature and do not have to undergo the cold chain storage systems like the current vaccines that are used to treat COVID-19. And with that, I would like to finish my talk and hand over to Heinrich, sorry.
Yeah, thank you very much for this very helpful study of lipid stability, which is always important to get better understanding. We can discuss that later on. We can now move on to the next speaker who is Dan Grommelin from Leiden in the Netherlands. He's known to everybody in the field, and she will also talk about an important topic addressing the cold reality of mRNA vaccine stability. So we really look forward to your presentation.
There is something not working. Can somebody help me?
It looks good. We see you, we hear you.
Oh. Yeah, but I don't
need to share, and then
I don't see my presentation. Do you see my presentation?
Not yet, so I think maybe it's a bit slow.
Can you share it through Zoom with a button share screen?
Oh, perfect.
Does it work?
Yes, you just now have to start the presentation mode and this. Oh, wonderful. This looks good.
Okay. Yeah, thanks for the invitation. Addressing the cold reality of mRNA vaccine stability is, or was, is the title of a presentation, of a publication that we published now a year ago, quite heavily cited. So it is an indication that apparently the topic that we discussed in this commentary, that it is picked up by others, and Heinrich already mentioned that, and also later speakers today referred to the stability issues that we still have to face. A year ago, we made a point that, and I cite here this quote, clearly the speed at which the RNA vaccine candidates have recently been developed prioritized optimization of in vivo delivery, in vivo expression, animal immunogenicity, safety results over storage stability. This is not a sort of traumatic impression of a frustrated person, but it was, I think, definitely a signal to the field. So having said that, that sort of sets the stage. And before going on, it's good to have some factual information about what are we talking about. And this is the situation the 5th of December, 2020. And of course, we are now 1.5 years later. So things have changed, and I refer to the EMA and FDA websites to sort of update the scheme here, but basically it's still the same. Stability in frozen conditions for months, stability in the refrigerator for days, and stability at room temperature for hours. So that is basically still the situation. And there's actually very little in the public domain that tells you about this stability in frozen state and stability in the refrigerator. There are some publications published on the stability at room temperature, and I quote two here. One is the publication by an Italian group from Paolo Minghetti, pre-drawn syringes of Comirnaty, so that's the Pfizer BioNTech vaccine for efficient COVID mass vaccination. And they look at three variables, syringe temperature, mechanical stress, and they have 3 quality attributes that they look at: the chain integrity and the size by dynamic light scattering and TEM. And the stability is, if you use it correctly, and 5 hours at room temperatures are okay, but shaking is really detrimental to the product. So shaking is something that should really be avoided. And that's what the protocol says. I mean, it's nothing new, but when they shook it, things really went wrong. So this is one. The second one is by a UK group, and basically they come to the same conclusion. So they also make the point that shaking and exposing the vaccine to excessive vibration causes aggregation. But that is what I could find until now on the use of the stability in use of the COVID vaccines, the mRNA COVID vaccines. Now, after 2.5 days, I would like to move now to sort of more the specifics of the stability of these mRNA COVID vaccines. And after 2.5 days of clean-on, this scheme should not be a surprise to you. It's taken from the Schoenmaker publication that was mentioned before. Ionizable cationic lipids, the helper lipids, and the PEG lipids. They are depicted here. For those of you who are really expert, you see that there's a mistake in it. I leave it for the time being. I'm not going to help you there. These are the lipids that we talk about. The preparation scheme, and this is a scheme that I took from Gerard Borgert. Also, after 2.5 days of clean-up, no surprise. And the preparation of what is called here liposome particles, I would call them LNPs, and sort of set them apart from liposomes, through microfluidics, an aqueous phase with the RNA, an organic phase with the lipids. They are mixed. You end up with the LNPs. The pH goes up from 4 to 7.4. And then there is this point, deep freezing. I mean, this is the basic point of my presentation. Why do we need this deep freezing? Why do we need deep freezing conditions? Can't we do something about that? And then you go to the literature, and of course the question is, what is the problem? And then the €1,000,000 question, why do we need these low temperatures and short storage times? It's the mRNA's instability, stupid, that is clear. But what became clear over the time, and it was mentioned by the previous speaker as well, is that lipids play a role by interacting with the mRNA and by determining the local pH. And this is something that needs some explanation. Michael already pointed out the excellent publication by Pecker from Moderna. And what she and others at Moderna figured out is that cationic lipids are readily oxidized and form lipids mRNA adducts. So the cationic lipids is oxidized, is hydrolyzed, an aldehyde is formed, and the aldehyde interacts with the mRNA, and the mRNA is inactivated. So the lipids play a role. It's not only the mRNA, but be aware of the lipids. And the second point is that what is the real pH inside the LNPs? Is it indeed 7.4 as the bulk pH, or are we talking about something between 4 and 7.4? There's still a debate there. Question mark, question mark, question mark. And last but not least, because of the surplus cationic charge in these LNPs, the surface pH is higher than the pH of the aqueous medium. And this is something that Klaas-Jan Zuidam and myself published already 25 years ago for cationic and negatively charged lipids, liposomes. And Wayman Steele and the Stanford group sort of confirmed that, and they estimate the pH inside the LNPs of around 9, pH around 9 instead of 7.4. And that has an impact, of course, on the degradation rate of the mRNA. And talking about this degradation rate, and I think I already showed this scheme already, but what happens with this mRNA, this single-stranded mRNA, and what is the chemical degradation? It can be oxidized, depurinated, deperminated, deaminated. Lipid adducts, I mean, this was not known until some time ago, like I said, with the Pecker paper. But the main degradation reaction that really, apart from the lipid adducts, but is the main driving force there is the hydrolysis. And this is shown on the scheme here that was taken from the Schoenach publication in 2000. Hydrolysis of the via the phosphate group, and this hydrolysis is dependent on the pH, both acidic and basic conditions, catalyze the degradation. So what can you do about that? And that is on the next slides. You can do something about the mRNA, and Michael already mentioned, no, before that, the speaker already mentioned that. You can look at the nucleosides, the modified mRNA there, using the standard pseudouridine or the 5-methyl-cytosine. They are supposedly, they bring in more stability. So that's one option. The other option is to look at the sequence. The GC motifs in the mRNA really improve stability. This is what CureVac championed. And structural UTR elements also helps in the stabilization. The GC motifs, they play an important role when you look at the secondary structure of the mRNA, and the more secondary structure you see, the more stability there is. So playing around with the sequence definitely helps stabilizing the mRNA. And last but not least, one thing is clear, that mRNA degrades and is inactivated by one break in the strand. So the shorter you make the strand, the less chance you have of breaking. And that is another sort of philosophy that you find in the literature. Shorten your chain length, and that helps. We go on to the options for mRNA LNPs that you find in the literature. And there is, first of all, biostabilization of the mRNA myself. Look at the previous slides. Selecting the nucleotides, GC content, the CureVac approach, increased secondary structure. Wayman Steele has an algorithm where they minimize the average unpaired probability of the nucleotides. So they really increase the secondary structure stability. Shorten the chain length, and that is championed by Zhao and collaborators. Then look at the purity of the cationic lipid. Packer, in her presentation, in an article on the lipid adducts, made it clear that a lot depends on the purity of the cationic lipid, so pay attention to that. Choice of the excipients, the buffer, of course, the pH, the osmolytics, cryoprotectants, lipoprotectants, you can play with those. And that, for instance, Pfizer BioNTech changed the buffer from phosphate to tris not so long ago, and that enhanced the stability and storage. Heinrich already mentioned freeze drying as an option. That's a logical option, and in particular when you consider that the EMA approved liposome freeze dried formulations for Ambizone, Markibo, and MEPAC. So that is definitely a way to go. And you see in the patent literature a lot of examples there, a lot of activity. And Zhao and Mutsu also published in the public domain, peer-reviewed articles. So there's a lot of activity there, and it looks like that is gonna work. It's energy intensive, and nowadays we have to be very careful with the energy. So spray drying may be a better alternative. There is some patent literature on that. I haven't seen any publication, a peer-reviewed publication, but there's patent literature on that. And last but not least, at the point in situ, at the bedside preparation of these mRNA lipids. Also, Heinrich gave an example there. There is more in the literature than just one. And I would like to spend some time in the last minutes on an example of the at the point in situ, at the bedside preparation of these mRNA lipid nanoparticles. And what they do is they pre-formulate off-the-shelf LNPs. And they call it LNCs, they're a little different, but basically it is LNPs. So they have them ready, no mRNA, and then at the point in situ, at the bedside, they mix that with the RNA, you get this complex. And the Erasmus group claims that they are active and stable. there's a lot to say about these publications, but I'll not do that. And after Erasmus, Kulkarni of the Colors Group published an example of siRNA, so not mRNA, but siRNA. And they did this, they played the same trick, but now with LNPs. Conclusions, stability first, that is a sort of phrase that is a little bit out of date now, but the rational design of optimally stabilized mRNA vaccine formulation should have top priority in the pharmaceutical development community. That's clear. I mean, that's the overarching change in paradigm that we need. And then it's very important to really look at systematic approaches and real good understanding of the key physical chemical degradation mechanisms. And we thought that we had, of course, understood everything, but the Moderna paper of Packer really showed us that no, we don't know, we still don't know everything. And this is a plea from an academic who consults for industry, of course, but academic, please publish everything in the public domain. Patents are a public domain, I know that, but they are not. They can't be compared to review publications in terms of their impact. Well, if you're interested in some more of this stuff, we published another paper later on, and then this is the Schoenmaker paper that really talks about in much more detail than the commentary about mRNA lipid nanoparticles, structure and stability. And with that, Heinrich, back to you.
Yeah, thank you very much. This was a great overview over all that stability issues, and I fully agree that these are complex and need to be followed up with further on. Also, I appreciate your remark of publication. This is very important, I think. But let's move on to the last speaker of that session, who is Hulot Hamal Al-Hamal, I hope I pronounced that correctly, and she will speak about mRNA siRNA combinatorial delivery to unblock immune checkpoint inhibition in solid tumors using stable nucleic acid nanoparticles, which is basically the older term for LNPs. So we look forward to your presentation.
Thank you very much, Haas. I'm gonna share my slides. So okay, so it's actually a great honor to speak after Dan, and I'm humbled, and actually that took me back to the very important physical chemical questions that people were more keen on, scientists were more keen to ask really those critical questions and not only exciting data to publish. So I will try to actually bridge our work with something related to issues to consider from the previous speakers, but also put this into translation and how do we care about the characterization of the material to link to the biological activity. So I'm Khulud Al-Jamal, and today I'm going to talk to you about using the LNPs, the new term, for co-delivery of different nucleic acid entities to exert different functions in a cancer cell. And the main purpose is really to induce cancer immunotherapy to treat solid tumors in cancer. So it's quite interesting how we kind of shifted from the classical way of using nanocarriers or drug delivery into combining it with other modalities. So I'll take you a little bit back to the journey when we started combining immunotherapy with chemotherapy. So chemoimmunotherapy. And one day I was approached by a clinician who was interested in redirecting very water-soluble drugs called biphosphonates. And one example was zoledronic acid, which we then switched to alendronic acid for other reasons. But the point was really, we would like to change the pharmacokinetic of the drug. So the interest was to shift the biphosphonates from the bone tissues and kidney excretion to solid tumors. And the reason for that was really to expand the gamma delta T cells in vivo. So rather than expanding the cells outside the body and inject them, if we can find ways to allow them to proliferate. And that's because it was known biphosphonates could induce the proliferations of gamma delta T cells. We've had a talk from Alberto yesterday, which actually gives even more insight about using these as just classical drugs, because they also seem to have other types of immune modulations. But then, not to spend long on this, we kind of, so we published a few papers to show the feasibility of using liposomal alendronate to increase the efficacy of gamma delta T cells to treat lung cancer model after intravenous administration. We then started combining chemotherapy and immunotherapy in a context of using drugs that are also immunomodulatory, such as doxorubicin, which is known to have an immunogenic cell death drug. And what that means is it induces cell apoptosis and death, but in a way that also activates the immune system, where our body recognizes the tumor as a foreign antigen. So it kind of vaccinates the body against tumor recurrence. And in this project, what we've done is we would like to co-deliver an immune adjuvant, in this case, the CpG, in a way that it will insert itself in the lipid membrane of the cancer cell, but at the same time deliver doxorubicin, which will then induce this apoptosis of the cells. And what you then would have is the cancer cells dying, releasing the apoptotic bodies, which is tagged with the immune adjuvant, that then drain into the adjacent lymph node and activate the T cells in a specific way against the tumor. So kind of really co-delivery, and this was done by intratumoral administration to have a high concentration of the doxorubicin. So we found actually combining chemotherapy with immunotherapy is promising. As you can see here from the tumor growth delay curve, you can see there is significantly reduced tumor volume when you have the combinations. And also there is increased number of the CD4, CD8 cells in the tumor. And that's statistically significant only when you have the combinatory treatment. So we know combining chemotherapy with immunotherapy is interesting on its own right. And coincided, fortunately or unfortunately, with what happened with COVID and the excitement using LNPs. We found ourselves actually shifting into applying LNPs to deliver those modalities. What we were doing at that time is using a cationic, not ionizable, but DOTAP containing a cationic formulation that would contain doxorubicin as an ICD drug at high concentration, but also we wanted to deliver an sRNA to potentiate the immunotherapeutic effect of doxorubicin. So what we've hypothesized then was these cancer cells have high level of expression of CD47, which is a do not eat me marker that stops macrophages from engulfing these cells. And then what we wanted to do is actually deliver an sRNA that would block or reduce the expression of CD47, and the doxorubicin would increase the expression of the corticosteroid, one of the markers for ICD, and that would reactivate the phagocyte activity in the tumor and lead to tumor elimination. So we've done 2 injections, and this was an intravenous treatment for the different treatment groups, whether it's an individual component or the combination. And what we found surprisingly is that when you give the SICD47 with the doxorubicin, you have very, very slow tumor growth. In fact, what we've got is 60% of the tumor cleared the tumor. There was no toxicity associated in terms of changes in body weight, and the spleen site, which is known to be enlarged when you have the CT26 tumor model, has actually remained within the normal range, suggesting low tumor load. We then looked at the immune population in the spleen to check if there is a systemic effect and then a tumoral effect. So what you can see here, the activated CD8 cells in the spleen, interferon gamma positive cells are significantly elevated in the spleen, and also the T cell population, CD4 and CD8, are much higher when you deliver this combination. We then prefer to completely move away from the chemo and stay with the immuno. So we thought we would synergize the approach by combining different modalities just by relying on the immune checkpoint inhibition. And then we decided also to switch from the DOTAP as a quaternary lipid into a cationic lipid into an ionizable lipid. So there was a slightly change with approach that coincided with everyone's interest. So in case of immune checkpoint inhibitors, I'm sure everyone knows, but there is an orchestra of immune checkpoint inhibitors. Some of them are immune stimulatory, so you would like to actually enforce these to activate the immune effector cells to attack the cancer cells, and some of them are inhibitory and you actually want to silence those. So the idea was, if we can develop a lipid nanoparticles that combine an siRNA that would do the silencing job with an mRNA that would increase the expression, whether it's on a T cell or a macrophage or a cancer cell, that's another choice for us to have, we could have a synergistic approach. So we would like to make these LNPs, and actually the challenge here is, can we load them with both entities in a same manner? You know, the size of mRNA is much higher than siRNA, so what are the implications in terms of loading and so on? And in terms of the site of action, they would both act at a cytosolic level and do the different job. There might be also molecular interactions in terms of one gene may affect the expression or the silencing of another gene. So all these questions we really wanted to address. So this was the work we published last year, and I'm going to share some of this data with you. So we've chosen two immune checkpoint inhibitors. The positive one was OX40. OX40 ligand is expressed on the cancer cell, and OX40 receptor is expressed on the T cells. We know we're not successful in transfecting the T cells, so at this stage we've actually remained interested in the cancer cells and the macrophage. And the idea is to have an OX40 ligand mRNA and a PD-L1 siRNA in the particles, and we went into that direction. So this is a nice cartoon of what we would like the LNPs to look like. And this is, we've used MC3 lipid. You could ask me why, I'll tell you it was just a choice, but now we're also exploring other type of ionizable lipids for comparison. And we've also used another PEG, which is a ceramide PEG, again, not a DMG PEG. So these are all things that we are investigating now, but here the point was really validating those targets. So this is how a TM would look like for the lipid nanoparticles, and what we see is no change in how they look like if they encapsulate siRNA or mRNA or both. So that's a good sign, but it doesn't mean you are encapsulating both when you have that competition. So in terms of size, they are below 150 nanometers, nice polydispersity, again, near neutral or slightly positive, and we've got above 80% loading efficiency by ribogreen. Now, whether this is all mRNA or siRNA, we don't know. So what we've done here is we've actually taken those LNPs, which could have a free component in there. We digested with RNase H, which should technically eliminate the external nucleic acid, and then we run the gel electrophoresis. And what we looked at here is the ratio between the siRNA and mRNA before encapsulation, so this is the mix, and in the dissociated lipid nanoparticles. And we could maintain the same ratio intensity, so we actually concluded that we are able to encapsulate both components. This is quite important when dosing an animal study. So we looked at the transfection efficiency here, and what you can see here is we want to ensure that we have biological activity of each component and in single and combination. So the cells are untransfected. You have a level of PD-L1. When you silence them, there is a silencing, and then when you combine this with OX40, you don't affect the silencing efficiency, but you express the OX40. So you see a shift in the OX40 and a reduction in the PD-L1. So this is just a robage tells you we were successful in expression and silencing simultaneously without compromise when the other component was there. The cell viability, obviously you get toxicity at high concentrations, but our tests were in the safe region here. And then I told you we are interested in cancer cell targeting, but also the macrophages. So these are J774 cells. We wanted to see the impact of the transfection on those macrophages. So the first thing we were surprised to see was when we incubate these macrophages with the LNPs containing the RNA, in the negative RNA, you see increase in the PD-L1 expression. So actually transfection do increase PD-L1 expression. And to mitigate that, we found that PD-L1, in addition to our original purpose, actually brings this down to the original level. So that was good. So we don't elevate the PD-L1 in the macrophages, but we are able to express OX40 ligand. So what we also found is we're able to activate these macrophages when they are transfused with the nucleic acids, and we can see higher level of CD40, CD80, CD86, even with the negative SNPs. So the SNPs or the LNPs on their own right have some immune modulation activity. We looked at the transfection in an animal model. Now, the question was, do we treat the mice intratumorally or intravenously? Of course, intravenously is more versatile. You can access different types of tumors, but we need to remember we're here at target validation stage, and we want to see positive results. So when you look at the transfection, you can see this is intratumoral transfection. These are the LNP signal, and this is the luciferase mRNA signal. And with the intravenous, you get liver signal, not surprisingly. And these are the ex vivo imaging, again, showing the effect mostly in the tumor, while intravenously you see liver and the spleen. So one reason is to avoid systemic side effects, but also to achieve high concentration intratumorally. We went with that route of administration. These are just quantitative data to the left, so I won't bother with that. But to the right, we looked at the distribution within the tumor cell population, but also other immune cells. So which cells are actually taking these LNPs? So the DID positive is the LNP signal. And what you can see here is actually B cells, T cells, dendritic cells are all taking up the particles, and the tumor cells, CD45 negative, and the non-tumor cells are also taking the particles. So there's a wide distribution in the tumor. In terms of quantity per cell, we know that DCs seem to like the particles, and actually the leukocytes versus non-leukocytes, you have much higher uptake in these leukocyte cells. In terms of tumor growth, we can see that the LNPs with negative cargo has an effect on the tumor reduction, but it was less obvious than the positive group. And we also saw, if you assess the immune cells in the tumor, you have higher CD4, not significantly higher CD8 in the tumor, but actually the tumor draining lymph nodes can see all the effect. So we can see that the CD4, CD8 cells are significantly higher in our positive treatment group, and also CD44 positive, a sign of memory or exposure to the tumor, and activated proportion is also higher. So that's indicating the LNPs are working, whether they are actively just working the tumor size or whether they are draining into the lymph node and causing the action, we still don't know. We then were asked by the reviewer to do a survival study. And I think that was very interesting because when we looked at the survival of the mice that were administered the particles intratumorally, we can see that about 40% of the tumor, the mice actually cleared the tumor. And we took these mice and we re-challenged them to see if there is a memory response. And we could see that actually they fail to form the tumor again, which means there's a memory response against the tumor. So we got 100% survival compared to the same naive mice of the same age, aged and re-administered. So this is what we think is happening. We are injecting the LNPs intratumorally. There are a range of cells, so you get these macrophages and the cancer cells. The LNPs transfect the macrophages. They express OX40 ligand that activate the CD8 cells. They, however, keep the PD-L1 level at a basal level. And when it comes to the cancer cell, we silence the PD-L1 and we increase the expression of OX40. Combining the effect together, we have a more effector function response that leads to tumor eradication and also creating a long-term memory against the tumor. Finally, I would like to thank the team involved. The work is mostly produced by Dr. Adam Walter and Hind Abdelbar and the rest of the team who helped in various bits, our collaborators on the early work I've shown you, and also the funder. And finally, I would like to thank you for staying with me till the end.
Yeah, thank you very much for this presentation. I think we are already a little bit over time, so I suggest that we move on to the debate room where all of you should see the link, and I invite you to go there, and then we can just follow up with the questions. Thank you, everybody.